Welcome to this explainer. Today we're going to absolutely shatter the illusion that plants are just, passive objects sitting in the dirt. We're diving straight into the shockingly dynamic, completely hidden life of everyday vegetables. I mean, think about it. Plants don't have eyes, they don't have nervous systems, yet they are constantly sensing their environment, calculating their resources, and literally making life or death decisions on the fly. And to understand just how complex this all gets, we actually don't need to look any further than your own kitchen.
Have you ever forgotten a potato in the back of your kitchen cupboard? I mean, we've all done it, right? You buy a bag, push one to the very back of a dark cabinet, and totally forget about it. But what you find weeks later, it isn't just a rotting vegetable. It's more like this alien creature with these long, pale, stubby shoots just reaching out into the void. Well, what's happening there isn't decay at all. It is an active, highly deliberate survival strategy playing out right there in the dark.
This weird phenomenon perfectly illustrates a concept called etiolation. Just look at the comparison here. On one hand, we've got the dark-grown potato shoot. It's ghostly, it's spindly, the leaves haven't even expanded. And on the other, your typical light-grown plant, sturdy with broad green leaves. So why the massive difference? Well, imagine a young potato plant sprouting underground in nature. It's in pitch-black darkness. If it tried to expand delicate green leaves down in the soil, they'd just get ripped to shreds. Plus, spending precious energy producing green chlorophyll when there's absolutely zero sunlight for photosynthesis, that would be a massive waste of limited resources. So the plant makes a ruthless economic decision. It allocates literally all of its energy into elongating those ghostly pale stems just to push up through the soil and find the light. It's essentially saving the really costly leaf expansion and chlorophyll production for later.
But here is the real question. What happens the exact moment that spindly little sprout finally breaks through and hits the sunlight? Bam! It triggers a profound set of morphological and physiological changes. This is known as de-etiolation, or more informally, greening. Almost instantly, that frantic stem elongation slows right down. Roots start to grow longer, the leaves unfold and expand, and the plant just kicks into high gear, rapidly producing chlorophyll. It is a massive, sweeping transformation.
Okay, let's dive into this. We're going to zoom all the way down to the microscopic level to really solve this mystery. How on earth does a plant cell actually know it's in the light, and how does it execute such a massive transformation? Well, it all comes down to three core stages of cell signal processing: reception, transduction, and response.
Let's start with step 1, signal reception. Because obviously, before the plant can do anything else, it has to actually see the light to know it's finally broken through the soil. Now, what's really fascinating here is the specialized protein that basically acts as the plant's eyes. It's called phytochrome. And unlike most receptors in a cell that are rigidly built into the outer plasma membrane, this specific light-detecting switch just floats right there in the cell's fluid, the cytoplasm. And guys, it is incredibly sensitive. We are talking about a receptor that can be triggered by the equivalent of just a few seconds of moonlight. Crazy, right? When a single photon of light hits this phytochrome, the protein actually changes shape. And that shape change, that is the vital first switch being flipped.
And scientists actually know for sure that this is the exact switch, all thanks to some really fascinating mutant plants, like the Aurea tomato mutant. Aurea means gold, by the way, because without the green chlorophyll, the plant's natural yellow and orange pigments become super visible. Now, this specific mutant naturally has severely reduced levels of phytochrome. So when scientists put it in the light, it barely greens up at all. But, and here's the cool part, when researchers took individual Aurea leaf cells and actually injected them with phytochrome taken from other plants, and then exposed them to light, boom, they successfully greened. It absolutely nailed the proof that phytochrome is the mandatory light-sensing mechanism.
All right, moving on to step 2, transduction, or as I like to call it, the relay race. Let's see exactly how this whole process builds from a tiny whisper of moonlight into a massive internal alarm bell for the plant.
Because that initial light signal hitting the phytochrome is so incredibly weak, the plant cell has to use what are called second messengers. Think of these as small molecules and ions inside the cell that act like a hyper-efficient molecular relay race. Their one and only job is to take that tiny initial signal from the receptor and just massively amplify it, broadcasting it loud and clear to the proteins that are actually going to carry out the physical response.
And the really crucial point here is just how dramatic this amplification actually is. When that phytochrome changes shape in the light, it instantly causes calcium channels in the cell to pop open. This leads to a massive, transient, 100-fold increase in cytosolic calcium levels. It's literally like someone whispering a secret into a microphone, and suddenly giant stadium speakers blast it out 100 times louder.
That is exactly what the calcium is doing inside the plant cell. But calcium isn't working alone here. That activated phytochrome also triggers an enzyme to produce another vital second messenger called cyclic GMP. And here's the kicker. Both that giant 100-fold calcium spike and the cyclic GMP are completely necessary for a full deethylation response. If you only have one of them, you're only getting a partial response. Together, they go on to activate different protein kinases, which you can think of them as the cell's specialized workers, perfectly setting the stage for the final act.
Which brings us to step 3, the cellular response. This is the moment where the plant takes this amplified two-pronged signal and actually starts physically altering its own growth and chemistry.
So this amplified signal acts like a super complex game of dominoes, and it uses two main mechanisms to get things done. First up, we have post-translational modification. This is basically a kinase cascade that runs around activating enzymes that are already just sitting dormant in the cell, waiting to be given a job. Second, and honestly perhaps even more powerful, is transcriptional regulation. The signals activate specific transcription factors that march right into the cell's nucleus, bind directly to the DNA, and literally start turning specific genes on or off. They can act as activators to ramp up new genes or as repressors to firmly shut down the old ones.
And this whole process brilliantly showcases the plant making remarkable, life-or-death economic decisions right down at the molecular level. Those transcription factors and activated enzymes, they immediately go to work. The plant ruthlessly turns off the genes that produce growth hormones like auxin and brassinosteroids. You know, those are the exact same hormones that were causing that spindly, pale stem stretching in the dark. And at the exact same time, it rapidly starts churning out the enzymes it desperately needs for chlorophyll production and photosynthesis. The stem stops stretching, the leaves unfold, the plant turns vibrant green. Basically, our forgotten pantry potato has successfully woken up.
Now that we've completely solved exactly how a little bit of light transforms a pantry potato from a pale, spindly stalk into a leafy green plant, let's zoom back out for a second. We really need to look at the macro implications of these biochemical pathways, because honestly, this is way more than just some quirky plant fact.
It is absolutely fascinating to realize that at the level of signal reception and transduction, the molecular machinery inside that humble potato is remarkably similar to the biology inside your own human body. Seriously, the similarities far outweigh the differences. When your cells need to respond to a hormone or some environmental stimulus, they use receptors, they use second messengers like calcium and cyclic GMP, and they use kinase cascades, exactly like our deethylating potato. The main difference? Plants respond by fundamentally altering their growth and development, whereas we might just respond by, moving out of the way. So I'm going to leave you with this final thought. Next time you're in the kitchen, take a much closer look at the fresh produce around you. When you inevitably find a sprouting potato in the very back of your cupboard, remember the incredibly complex microscopic relay race happening right inside it. It isn't just sitting there rotting away. It is an advanced organism actively calculating its next move, sensing the darkness, measuring its limited resources, and preparing to fundamentally reprogram its own DNA the very moment a single ray of light touches it. Thank you so much for joining me for this explainer, and remember, keep looking closer at the world around you.